I'd like to introduce Emily Remnant, who's going to tell us a little bit about her research that she's starting on looking for bee-specific chemicals to protect honeybees. So thank you, Emily, for taking your time to visit us and deliver your presentation. Over to you. Thank you.
Thank you so much, Barry, for the invitation, and thank you all for coming. It's really good to be here to tell you about some new research that we're currently just getting up and running. So it's early days. I will make sure that's clear at this point. First of all, I just wanted to let you know a bit about me. I'm a teaching and research academic at the University of Sydney. I've worked here for about 10 years. We have a big group full of happy students doing lab work and bee work and graduating and eating cakes. This cake was something I made for my PhD student, Amanda. It's meant to look like a honeybee pupae. I hope you notice the resemblance, and we're injecting it with viruses there, all in the name of research. But we have quite a diverse range of topics that we work on at the lab here, ranging from honeybee breeding, honeybee health and viruses, to native stingless bees and all of that. So we do a lot of different work here. And my research is really under these three categories, three Ps, I like to call it: pathogens, parasites, and pesticides. Most of my work is actually on these pathogens here and as how they apply to parasites, but I am also very interested in this third pesticide one, which I'll mostly be talking about today. So you'll all be aware of the pathogens that you might find in your hive. Some of those are quite common, and we have a lot of fungal and bacterial pathogens that are pretty common in Australia. But the main pathogen I work on are viruses. And so we know of at least 30 species of viruses that can infect honeybees throughout the world. Honeybees in Australia are commonly infected with 5 main viruses, yet we're still finding lots of other different viruses. And some of those ones that you might be familiar with are these two here, sackbrood and black queen cell virus. They occur at a pretty high frequency, even if you don't see symptoms. And my research largely looks at how the viruses of honeybees have changed since Varroa has spread. And that's a topic that I'm now going to hopefully explore as Varroa spreads around New South Wales. Now, the other area that I do look at a lot are the parasites of the beehive. So the main ones that we're affected by in Australia are the small hive beetle. This is the Athena timida, really big pest of Eastern Australia and a little bit of North and Western Australia. And obviously the parasitic mite Varroa destructor. So we've now had mites for a couple of years. And together it's sort of an interesting mix. We're now dealing with both parasites at the same time, and whether or not they might have synergy is a really important question as well. So we also have some emerging threats. You would have heard the recent incursion of Varroa jacobsoni in Brisbane. And we also have another invasive honeybee incursion in the north of Western Australia. That is the dwarf honeybee called Apis florea, and it has another mite that it's carried in as well called Varroa. So we always have that threat of further biosecurity incursions bringing in new parasites. And how do we best manage these hive parasites? So that's one area that my work looks into as well. The Varroa incursion, it's now in its second year since they were first detected. We've sort of gone through that eradication program, and we've now transitioned to management. And so I have been keeping a close eye on the spread, because back in the transition to management, we were located down here in Sydney. And I've been keeping an eye on the spread because I am very interested in collecting some samples and seeing how the viruses are changing throughout this zone. So now, as of a few weeks ago, my area of Sydney has been swallowed up by the Varroa invasion front. It's moving fairly slowly, I would say, considering that amount of time. It has sort of been on the verge of invading the Sydney Basin. To me, I think in the absence of some large-scale human-mediated movement, the natural movements of Varroa are sort of a steady spread southward. So I'm not sure how long it's going to take to get to you. I guess it remains to be seen. So the one thing I want to start off with, and it's relating to something that 2 of my students are working on at the moment, is what Varroa looks like. So you're probably all familiar with these sort of tan, orangey discs scuttling around on the back of bees. In reality, Varroa doesn't really look like that. There are quite a lot of adult mature females that have that appearance, but when you actually dig out a mite from a hive, it could be any range of these different colors. So these are the different life stages of the mite. And so when we're also looking for mites, we're looking in the honeycomb. It's easier to find the mites in the comb than it is actually on the back of an adult bee. And the other thing to look for, if you are getting down and sort of digging into your hive and trying to determine whether mites are there, they hardly ever hang out on the thorax. They're mostly found on this region of the abdomen and mostly on the left lateral bottom of the abdomen. So if you really want to find a mite attached to a bee, that's where to look for it. So I have two students, James and Rebecca, who are visiting Phil Lester's group at the Victoria University of Wellington. And they're currently collecting different life stages of mites. So here's a little mite family that they pulled out, taking out the pupae, collecting males, collecting foundress mites and females to analyze aspects of their genetics. All of this ultimately with the aim of identifying new and interesting ways for biological control of mites. So here's an experiment they were performing recently where they took adult mites and they took them out of the hive and they wanted to treat them with a particular RNA treatment. So looking at an RNA method of controlling mites. They basically soaked the mites in little test tubes for 24 hours in the fridge. So they take out these female mites, put them in a tube in the fridge. And then once they've had a good soak in overnight at 4 degrees, they place them on a pupae in these little capsules. And so they are being administered the treatment there. And so they'll monitor their survival and look at them over the next 48 hours. So that's something that we've got going at the moment. And I heard you're going to be hearing from Zoe Smeal next month, who recently submitted her PhD from this lab. And that will be a talk not to be missed for sure. So this gets me to the reproductive cycle of Varroa, and this is all a little bit related to my insecticide project, which I will get to, but I just wanted to mention a little bit about its life cycle. So basically we have on the left here a figure showing the honeybee brood cycle with our larvae, which are then capped, and then the pupae are developing, they darken over time, and once they've darkened and metamorphosis has occurred, they'll emerge as an adult. The mite females will detect the smell of the brood, and that is their cue to jump into the comb. And they do this before the capping occurs, and then they hide. Once that cap gets put on by the worker bees, they will feed on the side of the larvae. The female will then lay a single egg. It's their male, it's their son, and that egg hatches and it will lay, this is actually meant to say a female egg. It will lay a female egg every 30 to 70 hours thereafter. So once the females mature, they mate with their brother. And so this is a very inbred family here. And once the female bee emerges, only the adult females that are mature enough will survive. And on average, we're getting sort of a 1 to 3 survival. So we start with one female, and on average across the hive, due to the time frame, we get maybe 3 daughters coming out again. Now, one important point is that when the mites feed on the larvae and the pupae as they're developing, they actually ingest lots of nutrients, but they also ingest some very important hormones for the mites' development. One hormone that mites and bees use to help their development and help them to lay eggs is called ecdysone. So we'll be talking about that later. The other thing I want to note is if we don't have brood in the hive, for example, if you remove the queen or your hive goes queenless and there's a period of broodlessness, the mites don't have anywhere to reproduce because they are so tied to this reproductive cycle. And so that's a strategy that some people use to lower mite loads without chemicals, is to cage the queen or have a period of no brood in the hive. One other point to note is that I think we're currently in an interesting stage of Varroa management where the aim is really to slow the spread. We're not really looking in towards the long term just yet. So it's very short term minded. And so the current recommendations are to treat the hives with synthetic pesticides to get mite loads down, and that's to stop the spread from sort of getting out of control. And we're also in that education phase. So we're monitoring, reporting, recording, continuing to manage the biosecurity to prevent incursions, but then also now rolling out some education programs. A lot of people have mentioned their hesitancy to use synthetic pesticides, and we'll talk about that a bit too, because, we know that there's some negative aspects to them. I think they are really the advice being given in the short term, and I'm hoping that this is not Australia's long-term strategy. I think in the long term, we need to work towards a more integrated pest management approach. So some of you might already be aware of this. I'm certainly not an expert in the intricacies of integrated pest management. It's basically an overall philosophy of tolerating a pest's presence if it remains below a particular threshold where the farmer suffers economically or it causes financial damage. So tolerate your pest until it starts costing you money. And in general, it focuses on reducing chemical use and implementing other measures to keep mites low. When treating Varroa, this is a nice little infographic from the Bee Professor website. We have these sort of different options that you can go for. You can have the preventative options that discourage mite growth. So you could have a bee line that's resistant to mites and keep the mite levels low. You could also cage the queen and get rid of the brood, various sort of preventative measures. You can have some non-chemical methods. So that's mite trapping and brood break is there. Or you can start to use some natural and synthetic compounds. And so there are quite a few products available. And here they've noted that combining multiple methods definitely improves the efficacy. So if we expand that out into a bit more of a detailed and Varroa management pyramid, I've tried to summarize all the different parts of that framework here. And so generally we have down the bottom the least environmentally risky options, and gradually moving up to the more risky or costly options. So costly could actually be a financial cost, and that's considered a risk as well. So there are many different strategies that people have used to try to control Varroa. Not a single one of them has been 100% effective at this point in time. So they are all tools in the toolkit, but there's no silver bullet. And so we're kind of getting to a point where we still need to look for long-term solutions, because these current strategies are managing mites pretty well, depending on the situation, if you're using these strategies correctly. But there's nothing that can cure a hive of Varroa. You're always going to have Varroa there. So I'm going to go into a little bit more about the chemical side of things now. So currently we have two different categories of pesticides available. You would have heard of these as well, the organic and the synthetic pesticides. These are sometimes called soft and hard chemicals, the soft being the organic ones, hard being the synthetic. Those aren't necessarily representative of the chemicals because they have advantages and disadvantages, both of them. So some of them have higher efficacy, some of them have more safety for humans, some have undesirable residues left in the hive. So they all vary in their mode of action, and often their effectiveness relies on other factors in the hive at the time. So the mite loads, what stage of the brood cycle we're at, whether the temperatures are high or low. So there are lots of different considerations when deciding how to manage the mites in your hive if you're going for that chemical strategy. So to summarize what we're looking at throughout the world, there's 4 main categories of synthetic chemicals and 4 main categories of organic ones. This goes up and down depending on which manual you consult. But I will note that not all of these are currently available in Australia at present. I have tried to look up the ones that are mentioned on the DPI website. And so I've put an arrow next to those ones. Some of these other ones are used around the world elsewhere. So as part of a recent project, which I'll give a plug to at the end, we recently did a review of all the different chemical controls. And we found that in general, the efficacy of the synthetic products were considered to be higher than the organic products. However, with a caveat that that's when there's no mite resistance. So these chemicals are good until mite resistance develops. So that's where the mites can tolerate the chemicals. And one other thing to note is that in general, there were reports that these chemicals, if used incorrectly or at the wrong time of the brood cycle, are actually really quite harmful to bees. So there's nothing that's commonly used that's completely harmless to bees. So this gets me to our project. which is to think about pesticide use in a honeybee hive a bit differently. So I've recently, maybe the last 3 years, started working on this project. We've now received funding from Horticulture Innovation Australia, and we've also received very generous philanthropic donation, which kicked the project off to begin with from a donor to the University of Sydney. I work with these two biochemists, Professor John Mackay and Professor Ron Hill. Our goal is to develop safer selective pesticides that target Varroa and small hive beetles. And we're targeting a receptor called the ecdysone receptor. So it's a hormone receptor that I mentioned earlier, that molecule that Varroa needs to trigger its egg laying in development. So we're actually using a method called target-based drug discovery. Now, as a genetics person who works with insects, I know very little about the ins and outs of how you discover drugs because I don't really work in the pharmaceutical industry. But we're applying this in a new way to try and find a molecule. This is a chemical here that can harm our mite or our beetle, but be absolutely fine for the honeybee. So I'm going to try and describe that process a little bit. First of all, I just wanted to explain why we're following this route, and that is probably something that's fairly clear to most people who are working in agriculture, but maybe not so clear to everybody generally, that most pesticides that we use are broad spectrum, which means that they harm all types of insects, not just the pest you're trying to treat. And so you need to really carefully use those pesticides. You know, you're not going to treat a crop where you're about to take beehives. You need to time treatments really well. So that's the problem of broad spectrum chemicals. And now, how do they generally act? First, I want to start with one example, Fibronil. Now, this is a chemical that is really commonly used in agriculture and animal health. If you have a dog or cat, maybe check the box of your flea treatment. It could have maybe imidacloprid or fipronil. They're really common chemicals that are used for flea treatments. You might also be aware of fipronil for its other uses in the bee world. It was the chemical used in our feral bee baiting program that was employed during Varroa eradication. And it's also the active ingredient in the apithor beetle traps. So you can see here the back of a trap, it's got the fipronil active ingredient there. So seeing as this chemical is sort of very toxic to bees, it was the best chemical used for that feral bee baiting. We were interested in figuring out whether there's an alternative, a bee-safe chemical. I will note that if you're using apithor, generally there's no need to be concerned about your honeybees because the insecticide is safely contained in this plastic trap and only the beetles can access it. But you might have the unfortunate scenario of your hive flooding, and if the apithor trap takes on water, some of that chemical can leach out into the hive and it can cause some bee mortality. So considering that these chemicals are used in a beehive, we're thinking maybe we need to start thinking about some bee-safe alternatives. Now, a chemical, when it's acting on an insect or a pest, it is absorbed either through their hard shell, the cuticle, or via their stomach through the gut. And it might encounter some enzymes that can detoxify that chemical. But then some of that chemical will bind and interact with a protein, which we call the target site. So often that target site exists in the brain or the nervous system. And so some ion channels, which mediate neurotransmission, they can be bound to by a chemical and they cause neurotoxicity and death of the insect. Now, there's actually a really high rate of pests developing resistance to chemicals because when you're constantly exposed to this bad chemical, it selects for rapid evolution to become tolerant or resistant. So it's this whole adage of adapt or die. If you don't adapt, you will die. And so there are really strong selective pressure for species to evolve resistance. And there are mutations that can happen in the genes of the insect or the pest that provide an ability to tolerate that insecticide. So those resistance mutations can occur in the target site, so it can stop the insecticide from binding to the target, or they can occur in these detoxification enzymes, so it can allow the insect to better break down the chemical. And this resistance will be selected for and become widespread in a population because of its huge advantage. So since the introduction of chemical insecticides in the 1900s, the number of cases of insecticide resistance have been steadily rising over the decades. Here we have arrows indicating the introduction of different chemical classes, and we can just see it's really a cumulative occurrence of resistance developing. To spell it out, if we have a mixture of pests that have some resistance in purple, or they are susceptible or not resistant in orange, if you have a mixture of those individuals on a plant, you will apply the pesticide and the insects without resistance will die, these orange ones, leaving behind sort of resistant survivors in purple. Those resistant survivors will breed and multiply, and when the pesticide is applied again, they no longer work. And that's pretty much what happens with Varroa resistance too. So once it's in the population, it's very hard for it to be lost. As well as the fact that most pesticides are bad for bees, we are also very aware of the fact that resistance to Varroa's current chemicals is becoming hard to manage. So we are interested in developing new, safer insecticides. So we're targeting the hormone receptor, the ecdysone receptor. Now, these are really interesting receptors because they're not present at all in vertebrates, so they're safe for humans and other animals in the environment. They are specific to insects and mites, but they are different between different insect types. So there are small molecular differences that mean that they don't have that same mode of action, and so we can potentially select for different chemicals that act on one pest insect and not a beneficial. The method that we're using is structure-based drug design. So here we have our pest and our beneficial insect. We take the ecdysone receptor and we express it in the lab. So we basically make protein in a test tube. So I'll talk through a little bit about the process of how we do that as well. So we take our protein and we try and figure out what shape it is, its structure. So I just want to show you a hormone receptor. This is our ecdysone receptor here. It has all these little folds and binding pockets. If you are thinking about a protein receptor, this is to give you a sense of the size of that receptor. So here we have coffee bean, rice, and a sesame seed. So now I'm going to zoom in and show you a few different things. So we've got an amoeba there, we've got a skin cell there, we've got the egg cell, a sperm cell, a red blood cell. And if we keep going and zooming in, we're getting to the sort of micrometer scale. We're getting to bacteria. We're getting to a mitochondria, which is the powerhouse of the cell. We're going into the viruses, and then we're getting down to some small cellular molecules. So here we have an antibody here. So that's a protein, and hemoglobin is another protein. So our receptors that we're targeting, ecdysone receptors, are probably on the scale of some in this size here. And so the chemical molecules that bind to those receptors are more like on the scale of these sorts of molecules, these tiny molecules here. So if you zoom back out, there's, it's not something you can see at all with a naked eye. So when I say we look at the structure of the receptors, we're not seeing the receptors, we're seeing chemical signatures of what we expect them to look like. So here's one of our structures of the ecdysone receptor, and here is ecdysone bound to where the ecdysone hormone usually binds to our ecdysone receptor. So when ecdysone binds to this position, it causes this protein to trigger a lot of developmental processes in the insect. And so what we're trying to do now is find a chemical that binds in the same part that ecdysone binds to block ecdysone from doing its job. So if our new chemicals disrupt the function of the protein, they are considered to be a good drug or pesticide. Now, this method is how a lot of cancer drugs are actually found. So in human pharmaceuticals, you can take the protein target that you'd like to inhibit and do a screen where you find chemicals that bind to that protein and inhibit its function. So once we've determined the structure, we are also in parallel performing a chemical screen to find those small molecules that can actually bind to our ecdysone receptor. So this is ecdysone here. Now, there are many ways of going about this, and this is starting to get into the realms of not my area of expertise. So I'm going to try and explain it in the way I understand it. So what we have is a chemical panel where we have thousands and thousands of different chemical molecules that we take, and we try and see whether they can bind to our ecdysone receptor. And we have a special assay that tells us whether this chemical binds or it doesn't. We then get probably hundreds or tens of hundreds of different chemical hits. So I've just circled at random here what you might see in your results across a plate of different samples that you're testing of different chemicals. You might get 15 or 20. And these small molecules are something that we've identified that have some attraction or some binding ability to our ecdysone receptor. When you have those small molecules, you perform this process called chemical optimization. So these chemicals aren't the final pesticide that you'll end up using. They're just a little piece of the pesticide that you might end up with. You need to elaborate those chemicals, add different chains, and make them work better. And you want to make sure that they have strong binding affinity to your target and are also selective for your pest insect. So as I mentioned earlier, we have our ecdysone receptor from Varroa and honeybee. So what we want is for our chemicals to not bind to the honeybee one, but bind to the Varroa one. We want them to block Varroa's ecdysone receptor, but leave the bee's ecdysone receptor alone. So you go through this process of elaborating the chemical. And so that is something I don't really understand. So I'm going to use terms that I do understand. And being the mother of a 4-year-old, I'm choosing Lego. So let's say we start off with our small molecule that is this duck-shaped Lego here. The fragment elaboration involves taking another piece of Lego and attaching it to a part of the original molecule. So you might try a yellow piece here, you might try a red piece there, you might try a green piece here, or all of the above. And so these fragments are added to the side of your original small molecule to see if they work better. So once you've done that process, you want to make sure that you've got a chemical that in your lab looks like it's working against the mite, but not the bee. But then is it actually safe for a bee in real life when you test it in an actual insect in the hive? So we have a lot of toxicology, safety testing to be done. I will let you know we're currently at this stage here. So we've got our chemical screen up and running. We've found some small molecule hits, and now we're about to start this process of the Lego step, the chemical optimization and testing. And we're hoping to get a PhD student working with our chemist collaborators on this project soon. And once you've determined that you've got a safe product, you can then approach a chemical company for product development. Basically, I just wanted to summarize the key take-home messages. When looking at the chemical side of Varroa control, there is no current control method that is actually 100% effective. And so because of that resistance issue, we need to continually work towards developing new Varroa control methods. Relying on chemicals and a single chemical will lead to resistance development. And so things like chemical mixtures and rotations can help avoid that. But we also need to be flexible with our long-term solutions. So this drug discovery approach was something we thought could be explored. It's a new way of designing pesticides. It has not been done before, to our knowledge, in the pest insect world. But then there's also the other alternative, which I won't talk anymore on, is do we actually avoid the use of chemicals altogether? And that is via sort of breeding Varroa-resistant bees. So I'll just leave you with that thought there. Now, this is the report I mentioned earlier where we performed a review of the options available for transition to management in Varroa, an Agrifutures Australia report. It was led by Dr. Nadine Chapman and Dr. Michael Holmes, and then our contributors were John Roberts from CSIRO, Jody Gertz, Julia Grassel, and Sasha Mikhaev, and myself. And so our role was to take the information from international experts and apply it to Australia's diverse conditions. We've produced a report that is shortly available on Agrifutures' website, and it focused on these 5 areas of major concern. So we do have a treatment and management section, a breeding section, bee health and that sort of thing as well. So hopefully that will be available for people to download soon. So thank you so much for listening. I just wanted to plug some research I'm currently doing. Now, I've spoken to beekeepers clubs in Sydney lately, and so I had this message at the end. Obviously, I can't get mites from any of you, but I am also interested in sampling small hive beetles. So if you are interested in contributing samples or just connecting up and being a part of the research, please contact me, and here's my email address. And that is all from me.
Emily, on behalf of everyone here this evening, and also the people that are going to look at this recording in the future, we would like to thank you for your presentation.